So the diagnosis of MS remains a diagnosis of exclusion. So we don't have any tests that we can do to definitely say that somebody has MS or not. But we're now beginning to understand a lot more about the earliest stages of the disease. And we're constantly redefining the disease in terms of when it starts and what it consists of. So we used to, 20 years ago, require 2 clinical events, as our colleagues know well. But today, we don't require that anymore, and we are beginning to treat actually radiologically isolated syndrome. These are individuals who have MS-like lesions in their brain, but have no symptoms. And so, is that part of the disease? Well, a lot of us think so, and again, we're currently doing the studies to show whether treating them has an impact on long-term disability. So, in this effort to redefine the disease and when it starts, these molecular and cellular studies are becoming very important because these will play an important role in identifying individuals in the general population and also in high-risk groups like family members who are at high risk of developing MS themselves. So which one of these individuals are at highest risk to develop MS? Because either if we eventually have a preventive therapy, those individuals that are high risk are obviously the ones that should be prioritized for such treatments. And also, even if we don't have such a therapy, just identifying it as early as possible and intervening is going to be beneficial, for them. So then it begs the question, so what are the measurements that we need to make in people who are otherwise healthy to know that either they are at high risk and or have early evidence of disease? And actually Dr. Patsopoulos and I worked on this project where we have assembled data on over 45,000 MS patients and 65,000 control subjects to identify the genetic architecture of disease onset. And so that report is currently under review, should be published fairly soon this year. We're going to be reporting that there are over 234 genetic variations that contribute to the onset of the disease. There are more to be found, but that's a large number, and so we have now a pretty good idea of what the genetic susceptibility is for this disease. And actually, it relates more to other inflammatory diseases like lupus, type 1 diabetes, rheumatoid arthritis, also Crohn's disease. So these inflammatory diseases all share an architecture genetically, so people who are susceptible to one are also susceptible to others. But that's not sufficient. So I think the important message that the speakers spoke to is that any one dimension of information, any one measurement is not going to be sufficient. That we're going to need many different measurements and to bring them together. So there are 2 challenges. One is to find, of course, a collection of measurements that we need to measure, and exactly how many and what they are, we don't really know yet. The genetic information is certainly one of them. But it's a minor component, I think, at the end of the day. But it's an important foundation because, of course, genetics doesn't change as we get older and we encounter other environmental changes. So that's something that's easy to measure, and we can already bring that into the equation. And, of course, there's a lot known about the immunology of MS over the last 20 years. There have been tremendous advances in our understanding that have led to the treatments that we have today. Yet there are more recent advances that have been sort of even more fundamental in some ways, where we have used antibody-mediated data to define new diseases essentially, like neuromyelitis optica, and more recently anti-MOG disease as well. And so these 2 clinical entities share similarities with MS, but they behave very differently. And in the case of MOG, we're still trying to understand exactly how to treat it. NMO, has been around longer. We have better information. But even there, we still have, we're still working on developing treatments. But the important advance is that we were able to use antibodies to identify a subset of patients that behave very differently from the majority of MS patients.